So we're going to take a look at one of the things that you have to be able to do based on what we studied for amino acids. This is just a quick recap. Make sure that you can classify amino acids by characteristics like small or proline or negative or polar, aliphatic, aromatic. Just make sure that that's a skill that you're currently practicing. So today we're going to take a look at nucleic acids. This is the last of our primary metabolites. So a nucleic acid is a biopolymer that contains three types of monomer units. There's a heterocyclic aromatic amine base, either a purine or a pyrimidine. There's the sugar, the monosaccharide D-ribose or 2-deoxy-D-ribose. Then there's phosphoric acid. So the names and one-letter abbreviations you have to be familiar with. What you'll be given is a table that looks like this without the names. You have to be able to recognize the structure, identify what type of structure it is, and then the name of that structure. So if we take a look at a nucleoside, this is a building block of nucleic acids. It consists of D-ribose or 2-deoxy-D-ribose bonded to a heterocyclic aromatic amine base by a beta-glycosidic bond. So for example, if you take a look at uracil, think about drawing uridine, which includes the sugar. So this is the base plus the sugar. Again, if you need to, take a pause and then come back and check your answer. So a nucleoside is a building block of nucleic acids consisting of the D-ribose or 2-deoxy-D-ribose, depending on whether it is RNA or DNA, bonded to a heterocyclic aromatic amine base by a beta-glycosidic bond. So the beta-glycosidic bond is the anomeric bond, that's at the 1 carbon, connected to the base. This is our beta D-riboside. It's beta, again, because this bond is up. And then this is our base uracil. So if we add about the phosphate then, we need to think about where that phosphate will add. So again, press pause. Think about where you could form a bond to phosphate. So a nucleotide is a nucleoside that now has a molecule of phosphoric acid esterified with an OH of the monosaccharide. Typically, this occurs at either the 3' OH or the 5' OH. So if you take a look below, this is adenosine 5' monophosphate, or AMP, a very common cellular signaling molecule. Now, if you think about what you already know in terms of how we've named molecules like adenosine 5 monophosphate AMP, how would you name this compound? If you said ATP or adenosine triphosphate, you are correct. One of the things that you're going to have to be able to do is identify 5' and 3'. So think about how you would label your carbons and then identify 5' and 3'. That's important because the 5' phosphorylated end and the free 3' hydroxy end are one of the ways that we're able to identify what is the business end or what's the end of the DNA that's going to be continuing the molecule, or perhaps what is the end of DNA that forms the backbone facing outward. Deoxyribonucleic acids, or DNA, are a biopolymer that consists of a backbone of alternating units of 2-deoxy-D-ribose and phosphate, in which the 3' OH of 1,2-deoxy-D-ribose is joined by a phosphodiester bond to the 5' OH of another 2-deoxy-D-ribose unit. When we think about then primary and secondary structure, it makes sense that the sequence of bases along the pentose phosphodiester backbone of a DNA molecule, or an RNA molecule, will be read from the 5' to the 3' end. So our primary structure is the sequence of nucleotides. The secondary structure is the ordered arrangement of the nucleic acid strands. So a double helix is a type of secondary structure of DNA molecules in which 2 anti-parallel polynucleotide strands are coiled in a right-handed manner about the same axis. There are several properties of this secondary structure that are worthy of note. It's a thickness of approximately 2 nanometers. If you think about this pattern of the curving, that's going to occur over approximately 10 bases. There's a minor groove that's 1.2 nanometers and a major groove that's 2.2 nanometers. When we think about nucleic acids, we think of the double helix model of DNA secondary structure that was proposed by James Watson and Francis Crick in 1953. But the reality is the story is far more complicated than that. Watson and Crick's work was really built upon the work of at least 3 other individuals: Maurice Wilkins, Rosalind Franklin, and Linus Pauling, who's in this fabulous Hawaiian print shirt on the left-hand side. It was really Pauling's work in terms of hydrogen bonding, and then Rosalind Franklin's work in terms of X-ray diffraction of DNA and Maurice Wilkins' interpretation of that allowed for the understanding of DNA. So if we think about what that data would look like, it's an X-ray source that is applied to a DNA crystal, and then these diffracted beams are captured by photographic film. That's what created this image that you see here, which is what gave the clue as to the helical nature of DNA. Now, if we break that down and think about individual hydrogen bonds between bases, we need to recognize that within the pattern of DNA, there is what we call Chargaff's rule. So this is the hydrogen bonds between adenine and thymine, and the hydrogen bonds between cytosine and guanine. Note, adenine and thymine have 2 hydrogen bonds, whereas cytosine and guanine have 3. You're going to be responsible for recognizing how to draw the hydrogen bonds between a molecule of A and a molecule of T, as well as a molecule of C and a molecule of G. So part of that is recognizing what is the hydrogen bond donor and what is the hydrogen bond acceptor. The other piece of information that's really important here is to clarify a misconception. There's more than one type of DNA. There's bDNA. This is the predominant form in dilute aqueous solution. So this is a right-handed helix. It's 20 angstroms thick with 34 angstroms per 10 base pairs. The minor groove of 12 angstroms and a major groove of 22 angstroms. Then there's aDNA. This is a right-handed helix, but it's thicker than bDNA. It has 29 angstroms per 10 base pairs. And then there's zDNA, which is a left-handed double helix. One of the things that's important to recognize is that any model of DNA is going to have benefits and limitations depending on how that model is drawn. So for example, here, there's shallow groove and deep groove don't necessarily represent the scale that actually exists for bDNA. And you can't see the identity of the bases or the number of hydrogen bonds. They all look the same, but we know that they're not. So one of the things that you need to be able to recognize is when DNA is drawn poorly, right? When that model doesn't necessarily reflect the structure of DNA itself, or perhaps maybe when components of the DNA structure have been minimized for the sake of emphasizing some other piece of understanding. One of the things that we also have to think about for nucleic acids is their tertiary structure. So this is the 3-dimensional arrangement of all atoms of a nucleic acid, commonly referred to as supercoiling. We know that chromatin is DNA molecules that are wound around particles of histones in a bead-like structure. So if we think about our original structure of DNA, we then coil that around histones to form nucleosomes. Those nucleosomes are then packed around a protein rod, and then that chromatin forms loops. Those loops then are then coiled again, and it's the coil of the coil that forms our chromosome. That's a lot of packing. When we think about the chromosomes that exist for human beings, we have 23 pairs or 46 chromosomes. These are shown here. Notice the colored regions are applied color to help differentiate regions of alleles, right, or regions of genes that are close in proximity to each other. The other piece of DNA that is worth talking about is circular DNA. This is a type of double-stranded DNA in which the 5' and 3' ends of each strand are joined by a phosphodiester bond. Now, it's important here to recognize that we can have circular DNA that's relaxed. We can have slightly strained circular DNA where we have unwound twists, and then we have strained or supercoiled circular DNA. RNA are similar to DNA in that they too consist of long unbranched chains of nucleotides that are joined by phosphodiester bonds between the 3' OH of one pentose and the 5' OH of the next. However, the pentose unit in RNA is a beta-D-ribose rather than a beta-2-deoxy-D-ribose. And the pyrimidine bases in RNA are uracil and cytosine rather than thymine and cytosine. And RNA is single-stranded rather than double-stranded. It's important that you know these distinctions between RNA and DNA. There are several different types of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA. Notice that they vary in size, in the number of nucleotides, and in the percentage of cellular RNA. RNA molecules are classified according to their structure and function. So ribosomal RNA, or rRNA, is a ribonucleic acid that's found in ribosomes, the site of protein synthesis. Transfer RNA is a ribonucleic acid that carries a specific amino acid to the site of protein synthesis on ribosomes. And messenger RNA, or mRNA, is a ribonucleic acid that carries coded genetic information from DNA to the ribosomes for the synthesis of proteins. It's present in cells in relatively small amounts, and it's very short-lived. It's single-stranded, and the synthesis of mRNA is directed by information encoded on DNA. So a complementary strand of mRNA is synthesized along one strand of an unwound DNA starting from the 3' end. The synthesis of mRNA from dRNA is called transcription. Notice, right, we have anti-parallel strands. So we have our DNA template, and then we have our mRNA. The delightful news for you is that you are not responsible for knowing the codon table, but it is useful to have an understanding of how each codon encodes for a specific amino acid. So the properties of a code are that there's always 3 nucleotides that signal chain termination, that the code is degenerate, which means that several amino acids are coded for by more than one triplet. So leucine, serine, and arginine, for example, are each coded for by 6 triplets. And then for the 15 amino acids coded for by 2, 3, or 4 triplets, it's only the 3rd letter of the codon that varies. So glycine, for example, is coded for by GGA, GGG, GGC, and GGU. There's no ambiguity in the code. Each triplet codes for one and only one amino acid. When we think about nucleic acids, it's important that we understand that restriction endonuclease is an enzyme that catalyzes the hydrolysis of a particular phosphodiester bond within a DNA strand. There are over 1,000 endonucleases that have been isolated and their specificity is determined. Typically, they recognize a set sequence of nucleotides and they cleave the DNA at or near that particular sequence. For example, the cleaves are shown here below. Notice there are enzymes that cleave in a variety of different locations based on sequence. When we think about studying or understanding the structure of nucleic acids, one of the most common methods is polyacrylamide gel electrophoresis. This is a technique that's so sensitive that it's possible to separate nucleic acid fragments differing from one another in only a single nucleotide. We also can use chain termination or the dideoxy method. This is a method developed by Frederick Sanger for sequencing DNA molecules. The key to Sanger sequencing is the addition of a 2', 3' dideoxy nucleoside triphosphate, or DDNTP, to the synthesizing medium. This synthesis terminates at any point where a DDNTP becomes incorporated because there is no free 3' hydroxy from which the chain can grow. So if we think about what we know, typically there would be an OH here, but now instead there's a hydrogen, which means the chain cannot continue. The process of Sanger sequencing is shown here, where a variety of nucleotides are added, and then randomly the DD, in this case ATP, is added, and that terminates the chain sequence. Then it's possible to look at polyacrylamide gel electrophoresis of each of those fragments, and then the order of the fragments, from larger to smaller, is going to indicate the sequence then of the complement of the DNA template. The last part about nucleic acids that we're going to talk about is a genome is the total content of DNA in one virus or in one cell. The genome of viruses and bacteria contain one molecule of DNA. The molecule may range in length from 7,800 base pairs to 8 million base pairs. In eukaryote cells with nuclei, the genome is one cell's collection of chromosomes, each chromosome presumably consisting of one molecule of DNA. The human genome contains 23 chromosomes whose DNA lengths total about 3 × 10⁹ base pairs. To give you a sense of the variety here, this is the approximate DNA length of a variety of forms. Next time, we're going to talk briefly about lipids, and then we're going to start talking about metabolism. Thanks again. Have a great day.